Hey everyone, ever wondered how your cells get the energy to do? Well, everything. It all comes down to biochemical energy generation. Think of it like this: your cells are tiny power plants, constantly working to create energy. We're going to break down the 4 key steps in this fascinating process. Get ready to learn about how your body converts food into usable energy. First up, digestion. Just like you break down food into smaller bites, your body breaks down large molecules, carbohydrates, fats, and proteins, into smaller, more manageable pieces like glucose, fatty acids, and amino acids. Enzymes are the unsung heroes here, acting like tiny scissors. Enzymes work by binding to specific substrates and catalyzing reactions, speeding up the breakdown process without being consumed themselves. For example, glycolysis converts glucose into pyruvate, yielding a net gain of 2 ATP and 2 NADH molecules. This process involves several enzymatic steps that break down the 6-carbon glucose into 2 3-carbon pyruvate molecules, producing energy in the form of ATP and electron carriers, NADH. Key takeaways. Digestion breaks down large biomolecules into smaller units. Enzymes catalyze these reactions efficiently. Glycolysis produces 2 ATP and 2 NADH per glucose, setting the stage for further energy production. Now, for the main event, the citric acid cycle, also known as the Krebs cycle or TCA cycle. This is where the magic really happens in cellular energy production. Acetyl-CoA, our central hub molecule from the previous step, enters this cycle. It combines with oxaloacetate to form citrate, kicking off the process. Through a series of 8 chemical reactions, the acetyl group from acetyl-CoA gets completely oxidized. This produces carbon dioxide, reduced coenzymes NADH and FADH₂, and a molecule of GTP, which is equivalent to ATP. For each acetyl-CoA that enters, we get 3 NADH, 1 FADH₂, 1 GTP, and 2 CO2 molecules. The beauty of this cycle is that it's truly cyclical. At the end, oxaloacetate is regenerated, ready to pick up another acetyl-CoA and keep the energy production going. It's like a merry-go-round that never stops as long as there's fuel to burn. Key takeaways. The citric acid cycle completely oxidizes acetyl-CoA, producing important energy carriers like NADH, FADH₂, and GTP. It also releases carbon dioxide and regenerates oxaloacetate to keep the cycle turning. This process is crucial for generating the energy our cells need to function. Now we reach the final step, the Electron Transport Chain, or ETC. This is where the energy from NADH and FADH₂ produced in the citric acid cycle is finally converted into ATP. The ETC is a series of protein complexes embedded in the inner mitochondrial membrane. Electrons from NADH and FADH₂ are passed along this chain, releasing energy that pumps protons across the membrane. Let's look at the ETC in action. NADH and FADH₂ donate their electrons to the chain. These electrons travel through complexes O through YV, while protons are pumped out, creating a gradient. NADH delivers electrons at complex I, while FADH₂ starts at complex II. The electrons move down the chain, losing energy at each step. The pumped protons create a gradient across the membrane. This gradient powers ATP synthase to make ATP from ADP and phosphate in a process called oxidative phosphorylation. ATP synthase uses the proton flow to synthesize ATP. This yields about 10 to 12 ATP molecules per acetyl-CoA oxidized through the CAC and ETC. To wrap up, the electron transport chain harnesses electrons from NADH and FADH₂ to create a proton gradient. ATP synthase then uses this gradient to produce ATP through oxidative phosphorylation, generating about 10 to 12 ATP molecules per acetyl-CoA. Let's talk about why Gibbs Free Energy, or Del-G, is so crucial in cellular respiration. All the steps we've seen are controlled to make sure the overall process has a negative Del-G, meaning it releases energy and happens spontaneously. Del-G tells us if a reaction will happen on its own. Exergonic reactions have negative Del-G, they release energy. Endergonic reactions have positive Del-G, they need energy input. The cell cleverly couples these reactions. An exergonic reaction provides energy for an endergonic one, like breaking down glucose to make ATP for building proteins. This keeps the total Del-G negative, so everything works. Without this careful control, the process wouldn't be spontaneous and the cell couldn't survive. Key takeaways. Negative Del-G means spontaneous energy release, and coupling drives essential functions like synthesis and movement. Let's say you're lifting weights. Your muscles need a ton of ATP to contract and allow you to lift the weight. This ATP comes from the biochemical energy generation process we just discussed. Glucose is broken down, goes through glycolysis, then enters the citric acid cycle and the ETC, ultimately providing the ATP needed. ATP powers muscle contraction through the sliding filament model. ATP binds to myosin heads, allowing them to detach from actin and prepare for the next contraction cycle. This energy process works for all types of muscle contractions: concentric when you lift weights, eccentric when you lower them, and isometric when you hold them steady. Key takeaways: ATP from cellular respiration powers all muscle contractions, whether you're lifting weights, running, or jumping. The process starts with glucose breakdown and ends with energy for action. So there you have it. Cells create energy in a carefully orchestrated series of steps involving breaking down molecules, creating acetyl-CoA, running the citric acid cycle, and using the electron transport chain to produce ATP. Remember, each step is crucial and they all work together to power your body. Exciting research continues to improve our understanding of how cells create energy. And it's not just for the body, but for things such as creating alternative fuels. Future trends include CRISPR for mitochondrial editing, synthetic biology for biofuels, personalized medicine for energy disorders, and AI modeling of metabolic pathways. Hope you enjoyed this biochemical journey.